Malaria, a deadly infectious disease caused by the Plasmodium parasite, continues to pose a significant global health threat. While various factors contribute to the susceptibility of individuals to malaria, recent studies have shed light on a specific genetic component, the AA genotype. This video explores the scientific evidence linking the AA genotype to an increased susceptibility to malaria, delving into the genetic basis, epidemiological implications, and potential avenues for targeted interventions. The AA genotype refers to a specific genetic makeup, usually involving a pair of adenine bases in a particular gene associated with susceptibility to malaria. Genetic research has identified specific polymorphisms in genes such as HBB, HbA1, and others, contributing to an individual's genetic predisposition to malaria. Genetic basis of susceptibility. 1 hemoglobin variants. The HBB gene encodes beta-globin, a crucial component of hemoglobin. Specific variants associated with the AA genotype may result in altered hemoglobin structure, creating an environment conducive to Plasmodium falciparum, the most deadly of the malaria parasites. 2. Immune response. The AA genotype has been linked to variations in immune response mechanisms, affecting an individual's ability to combat Plasmodium infection effectively. Epidemiological evidence. 1. Global distribution epidemiological studies have demonstrated a higher prevalence of the AA genotype in regions where malaria is endemic, such as sub-Saharan Africa and parts of Southeast Asia. 2. Association with severe malaria. Research indicates that individuals with the AA genotype are more prone to severe forms of malaria, including cerebral malaria, which can have life-threatening consequences. Malaria endemic regions. Populations in malaria endemic regions exhibit a higher frequency of the AA genotype, suggesting an evolutionary connection between genetic susceptibility and the prevalence of malaria. Potential implications for public health. 1. Targeted screening. Identification of individuals with the AA genotype could facilitate targeted screening and preventive measures in malaria endemic regions. 2. Genetic counseling. Genetic counseling services may play a role in educating individuals about their genetic predisposition to malaria and the importance of preventive measures. 3. Drug development. Understanding the genetic basis of susceptibility may contribute to the development of targeted therapies and vaccines. Studies unequivocally point to a genetic link between the AA genotype and an increased susceptibility to malaria. This knowledge not only advances our understanding of the complex interactions between genetics and infectious diseases, but also holds potential implications for public health strategies in malaria endemic regions. Further research is essential to explore the full spectrum of genetic factors influencing malaria susceptibility and to develop effective interventions that can mitigate the impact of this devastating disease on vulnerable populations.